This will be the last sexual differentiation lecture, so we're going to talk about some interesting things that can happen along the way. So remember, we really had gotten a good picture of the indifferent fetal sex, the bipotential gonad, the differentiated fetal gonadal sex. We mentioned just briefly about estrogens and testosterones influencing development there. But we're going to back up and talk a little bit about chromosomal sex and how that gets some of this process started. We certainly know a lot more today than we have when anatomists were first starting out. For example, Vesalius, he was an anatomist of the 1500s. Why am I bringing him up? Because he is hailed as being this great anatomist. However, he really had trouble getting the female reproductive system down. He was convinced that female reproductive structures were just poorly developed male structures. Here's some of his original sketches. Some of them look like inside-out penises. And the problem with what some of these, seminal papers and ideas and things that have come from these historical figures in the literature is indeed that we really have to change the way that we think. So some of that really is pervasive in the literature. So say it with me now. Female is not a default developmental pathway. Okay? Making ovaries, making testes, making these other structures, it's an active process that requires not just the presence or absence of a hormone, but a really clear developmental program. So the discovery of what is called the SRY gene really actually did as much damage, I think, as it did good with our thinking about development. Because a lot of times when you pick up a textbook now, like your Martini and Nath Fundamentals of Anatomy and Physiology, sometimes it'll say sexual differentiation and then they'll just talk about male development and there really isn't enough on female. And it was the discovery of the SRY gene, which is called sex reversal on the Y. So it's on the Y chromosome. So we know that the Y chromosome determines maleness. So it was the discovery of that that gave us a little bit of a clue into how we get from a chromosome to a gonad to a fully differentiated structure. So what was put out there is, oh, well, if this SRY gene is present on the Y chromosome, we're going to get a male. If it's not, it's female. Again, giving kind of a nod to that, oh, it's no effort to become female. It's the default pathway. This is false. Scientists just hadn't really had enough time to dig deep enough. I think your Sex and Gender book does a really great job in Chapter 3, walking us through a complex developmental program. I highly recommend that you read that because they do a really good job with it. And it's brought down to the level where you don't have to know really complex genetics. I'm just going to highlight a couple of things and give you the punchline here. But again, go back and read it one more time in the text. So we'll just orient ourselves to the figure. Again, we've got our bipotential gonad here. And then all of these things that just look like alphabet soup at this point, these are genes or proteins that could be produced. And it's a really coordinated effort, and not just having or lacking SRY, but there's all of this stuff that happens downstream that either needs to be present or absent to indeed get a testes or to get an ovary. So again, just punchline things here. It's not just having SRY, but we also, with the SRY, need something called SOX9. Again, don't worry about their funny names. A lot of this stuff was figured out in fruit flies, so they all have some silly names. So if we have a SOX9 and if we have SRY, then yay, we'll go down this path and make a testes. If we don't have SRY, right, and we don't have SOX9, that doesn't automatically get us an ovary. And I think this is where a lot of the information was lacking up until the time. It is possible for those external structures, the genital tubercle, the labioscrotal folds, those are going to end up looking female perhaps without the influence of dihydrotestosterone, but you will not get an ovary if you simply just lack SRY and SOX9. We need to go down this road and have these other proteins and genes turned on. Something called R-spondin seems to be the master regulator in terms of creating the ovary. So it's likely not just SRY or no SRY. That's not the story anymore. It's a lot more complicated. So punchline from that paper, SRY plus SOX9 gets you a testy. No SRY, but you need R-spondin to get an ovary and to develop female. So this is a very active and complicated process. Okay, one other thing I want to mention too, again, we'll get more into chromosomes and sex determination in that aspect later. But we know, right, that a sperm cell, right, this guy over here, okay, each one of these is either going to carry an X chromosome, right, or a Y chromosome. Okay. I'm sorry, that's an egg, right? So the egg will be X, the sperm will either carry the X or the sperm carries the Y chromosome. So there is one case where we can get the XX male. So that's actually really interesting. What we haven't talked about in terms of how chromosomes replicate and get divided up into sperm cells and egg cells, but we will get into that later. So whenever we have chromosomes lining up and getting ready to divide, what can happen is they'll undergo this phenomenon called crossing over. So let's actually switch here. So in the process of making sperm and egg cells, chromosomes will line up from male and that came from male and came from female. So this is very gender conforming here. So assume the blue chromosome came from dad and the pink chromosome came from mom. And we're getting ready to pull them apart and make sperm or egg cells. All chromosomes, regardless of whether they're X, Y, 1, 2, 3, 4, they can do this thing called crossing over. So right before we make sperm and egg cells, these guys can get very close to each other and little bits of them will pinch off and swap. So now you've got a little bit of pink chromosome on your blue chromosome and a little bit of the opposite as well. Okay. If this indeed happens between an X and a Y, it is possible, right, to take the SRY gene off of a Y chromosome, cross it over, and stick it onto an X. So now we have an X that's carrying SRY. If that X chromosome is the one chromosome, or if that X chromosome is carried by the sperm cell that combines with the egg, it is possible to get an XX male because you would have SRY, you'd presumably have SOX9 since that's on a completely different chromosome, and you would go down that testes developmental pathway, and then everything would appear male about that person, except for if they had a blood test. If they had a blood test, their cells would say XX, which without close look at the sequence looking for SRY, that would raise some flags and cause some questions. So it is possible at the chromosomal level for your chromosomes to not match your sex and to not match your gender identity, which we'll get into gender much later.